We're going to be talking about cell division, but in order to understand what happens during cell division, we first have to understand some terminology that is around chromosomes. So in sexually reproducing organisms like ourselves, chromosomes typically occur in sets. So for example, humans have 2 sets of 23, so that's a total of 46 chromosomes, but again in 2 sets. When an organism like us has 2 sets of chromosomes, we are referred to as diploid, so di for 2, or 2N. And in diploid organisms that sexually reproduce, each set is inherited from the parent. So an egg cell has 1 set and a sperm has 1 set. These are called haploid cells, haploid or 1N. So if you have a haploid egg and it gets fertilized by a haploid sperm, that's going to get you back to the diploid zygote, which is the fertilized egg, and that will eventually become, of course, the diploid adult. So this cycle of having haploid cells that combine together to form a diploid cell is part of the sexual reproductive process. So when an organism or a cell is diploid, each member of the chromosome set has a matching chromosome in the other set, and these are called homologs. So let me draw out what I mean. So you have a chromosome 1. So this is chromosome 1. And you got 1 from your mother. So this one came from your mother, or the egg. And then you've also got an exactly the same or very, very similar chromosome 1 that came from your father. These are homologs, or they are homologous. So homologous chromosomes are matching chromosomes. The information in those chromosomes is almost identical. Very, very small differences lead to the differences in traits that we think of as inheriting from each parent. And so if you went through the majority of your chromosomes, you would see that you always have 2. One that you inherited from your mother and one from your father. So this might be chromosome 2. Then you might have chromosome 3. So the chromosomes differ in size, as you can see that I'm sort of indicating here, but you always have 2. One you inherited from your mother, one you inherited from your father, and those 2 homologs are always virtually identical. The only exception to this rule is the sex chromosomes. In men, they have 2 different ones. They have an X, which is big, and a Y, which carries totally different information. It does not match. So the sex chromosomes, the X and the Y, are not homologs. That is the only exception to having homologous pairs. So this is also called a homologous pair. We're going to use that term a lot in this chapter. So this is a homologous pair, this is a homologous pair, this is a homologous pair. A karyotype is the way to display the chromosomes that an individual has. So this is one way to test someone for a chromosomal abnormality, is to take their cells and treat them in a specific way, and then look at the chromosomes that are present. And then they go through and they match them up and create a karyotype. So this isn't how they are in your cell. Somebody had to go through with a computer and move them together to make them line up. So they put the 1 next to the 1, the 2 next to the 2. So this is a homologous pair, this is a homologous pair, homologous pair, homologous pair, homologous pair, and so on. And the chromosomes are numbered from largest to smallest, so 1, 2, 3, 4, 5, so on and so forth to 22. So you have 22 autosomes. So these are these ones that occur in homologous pairs. Every human also has the 2 sex chromosomes. A female has 2 X chromosomes, a male has an X and a Y. So in this case, this karyotype is showing that this is a male. There's an X chromosome and a Y chromosome. So this is the karyotype of a normal male. So chromosomes are going to be central to the cell cycle and cell division. And so we can get a little bit of a preview of this in that when a cell is first born, if you will, or when it's first made, it'll start its sort of life cycle by growing. It'll just get bigger, it'll make stuff. Then the next step is that it's going to copy the chromosomes. So here's the one chromosome. And just remembering and linking back to what we saw in chapter 11, this is a DNA double helix. When the DNA gets copied, it's going to make 2 copies of that same chromosome. So now we see 2. And what that means, and again sort of connecting back to what we were looking at before, is I now have 2 double helices. They actually stay attached to each other in a position called the centromere. So that's called the centromere. And each one of these is a sister chromatid. So sister chromatids are not really sisters unless you think of them as identical twins, okay? They are exact copies of each other because it's 2 of these. But when they're copied and held together like this, they're sister chromatids. And again, reminding you that this is 2 double helices that are still attached to the centromere. So then the cell will divide some more, or rather, sorry, it'll grow some more after it's copied its chromosomes, and then eventually, in many cells anyway, it's going to divide the chromosomes. So these 2 sister chromatids that are together will separate, and now I have 2 separate chromosomes in 2 separate cells. And then here we see the connected 2 double helices. Now each one of these double helixes will go to the new cell. So the cell will grow. Then it copies its chromosomes and makes 2 sister chromatids, or 2 double helices, and then the cell will divide those chromosomes and divide into 2 cells. And so that's the basic process of the cell cycle and cell division.